Hello, I'm Jessilyn Dunn. I'm an assistant professor at Duke University in the departments of biomedical engineering and biostatistics and bioinformatics. I'm the director of the Big Ideas Lab at Duke, which is the biomedical informatics group integrating data engineering and analytics. I'm very grateful to Ken Bolin and Peter Mucha of UNC for the invitation to give this didactic seminar for the SAMC program on data science in the social and behavioral sciences, or the DS-SBS program, which is supported by the National Science Foundation under the grants listed here on this main page. Today, I will be giving a seminar on the digital data from wearable technology. So we'll talk about many different aspects of digital data and how it can be handled for analysis to improve healthcare. So to start out, I have a bit of a puzzle that I like to put up here on the screen where we have 8,760 small blue squares on the screen. So each of these blue squares represents hours in a year. So in total, 8,760 hours are in this year. And I want you to, for a moment, pause and think about how many of these squares represents what you think is the average amount of time a person spends with a physician during a year. So this was, prior to the pandemic, and based on this year, things are probably quite different. But in the past, I think it's helpful for us to think about what proportion of the time we're actually with a physician thinking about our health. So think about whether you might think maybe 5 or more squares, or 2 squares, or 1 square. And then we'll flip over to the next slide, where we can see that actually the average person spends only 15 to 20 minutes with a physician each year. So that's the time most people spend actively thinking about their health and the amount of time a physician has to gain a full picture of a person's health status. The discrete and sparsely sampled clinical data are an incredibly poor representation of overall health. Luckily, we have these new technologies that are making it possible for us to change this paradigm to better capture the complexities of human health. So through precision medicine and wearables data, we can continuously monitor health to arrive at preventative and actionable health insights. Using digital health methods, we can turn a 15 to 20 minute snapshot of a person's health into a true summary of current and past health, taking into account all of the relevant information, giving a full picture of health both inside and outside of the clinic. So why does this matter? Why do we need to have information about somebody's health outside of the clinic? Well, we've seen in these little squares that there's a large proportion of time when health is occurring, but it's not being monitored. And one of the examples that we have of a reason why it's so important to be able to monitor health continuously is the detection of arrhythmias. So arrhythmias are a condition that can result in heart attacks if untreated, and ultimately can result in death. And they're very straightforward to detect and treat if they occur when a person is in the clinic, or if they are detected through some sort of external monitoring. But in the past, the external monitoring has only been for a short period of time, something like 48 hours. And that in and of itself was an advancement over previous technologies that were only used to measure ECG in the clinic itself. So the benefit of these low-cost consumer-grade wearable devices, like what we might think of as a smartwatch from Fitbit or Apple or Garmin, enables us to detect continuous information about a person's health so that we can detect clinically relevant events outside of the clinic. And arrhythmias is the perfect explanation of this, because oftentimes arrhythmias can occur rarely and infrequently, and if they don't occur in the 48 hours of the external clinic monitoring, then they would never be caught. And so this continuous and non-invasive, very easy to use monitoring allows us to be able to detect things like arrhythmias outside of the clinic to enable treatment before any sort of adverse cardiac event occurs. And so one of the most common examples of this today that we see is the Apple Watch algorithm for arrhythmia detection. And so in December of 2018, Apple published this white paper about their arrhythmia detection algorithms, discussing both the PPG-based detection, which is one sensor that is used to measure heart rate, and another type of sensor on the more later stage devices from the Apple Watch Series 4 and on that actually uses an ECG-like measurement. And so later in this talk, I will dive into the details of what PPG is, or photoapplied tomography, and what ECG is, or electrocardiography, and why these different types of measurements matter and what they can be used for. But here, I think something that's really important, particularly for quantitative scientists to think about, is that the measurements that are taken from these wearable devices are often input into a proprietary algorithm by companies. So often, as users of the technologies, all we see is an output that tells us whether or not an arrhythmia is likely to have occurred. But we know as quantitative scientists that under the hood, there's a lot going on, from the collection of the primary digital data to the calculations that determine whether or not measurements are likely to be arrhythmias or not. And so we're going to talk a little bit about what goes on under the hood of these proprietary algorithms, so that if we want to move toward a world that has more open and visualizable algorithms for what we call digital biomarkers, or methods to detect health and disease from digital data, we will be able to move toward that world. So with that motivation in mind, that we have some of these infrequent but very important health events that, if detected, could lead to preventative care, this talk today is designed to cover a range of topics, from an overview of wearables data and how we access that data, what the different types of wearables data are and how we capture them, how the data can be used and validated. And so again, we'll talk in that section about digital biomarkers. What is a digital biomarker? How can it be used in practice, and how can we develop them? And leading to that development is understanding how to extract useful knowledge from wearables data, given that the data that we can collect now is much higher resolution, more longitudinal, and is just traditionally different than a lot of the data that biomedical scientists are used to handling. I'll talk a little bit then about research topics and using these different types of wearables data, and I'll discuss some ongoing efforts in a specific research area to detect digital biomarkers of influenza-like illness and COVID-19, which I think has become increasingly relevant in the age in which we're living today. And then finally, I will briefly touch upon the future of digital health, and we'll talk about where we see the future going. And with this, I would like to thank my PhD student, Peter Cho, who helped me to arrange this presentation and to organize this agenda in a way that is hopefully exciting and interesting and digestible to a range of people from varying backgrounds. So with that, we'll dive into an overview of wearables data. One of the huge benefits of using data from wearable devices is that wearables are becoming ubiquitous. So these started as devices in the fitness industry that were of interest to people for tracking step counts, for example. And as entertainment value on these devices increased, for example, the ability to text message and make phone calls and read articles on smart devices, people began purchasing more and more of these devices. And as the devices' sales increased, the number of devices that actually contain a heart rate monitor of some kind also increased. And so one of the reasons, aside from just the entertainment value for the common person, is that there are a lot of benefits of the ability, as we discussed earlier, to be able to detect health changes from devices that are far less invasive than they once were. So one example that we're looking at here is the example of the Holter monitor. And so we talked earlier about this idea that in order to detect arrhythmias, once it was necessary to wear a device that looked like this, that was pretty inconvenient to use and could be worn for roughly 24 to 48 hours to detect arrhythmias. And there are newer devices, for example, the Zeopatch shown here, that enable monitoring of ECG for longer, for up to 14 days. But again, this is not as convenient as a device that sits on your wrist that can be removed easily for showering, and that is able to continuously monitor without you noticing that it's there. And the same goes for devices that are used for people with particular types of diseases, for example, type 1 or type 2 diabetes, where individuals have to continuously monitor their glucose. And oftentimes what is used is a finger prick. And so you can see here this figure on the left showing a person taking a prick of blood from their finger and measuring their blood glucose, to a wearable device on the right, which is a continuous glucose monitor, which actually uses a thin fiber inserted under the skin and is able to continuously monitor blood glucose daily for up to usually 2 weeks, and that device can be replaced. And so this allows for not only a more convenient method of monitoring, but also a more continuous set of measurements. So why are wearables useful not for the individual, but for researchers broadly? Well, because we have so many people wearing so many different types of devices, this enables us to do some population level health monitoring. So one of the major benefits in having so many individuals using wearable devices is that it allows us to measure health changes on a population scale. And this has become increasingly relevant during the time of COVID-19, where our goal is to understand where there are outbreaks and hotspots. And so this is one example of a study done by Evidation Health, where they were comparing the changes in behavior and physiology for people who have various types of influenza-like illnesses. So one of these was a pre-COVID-19 influenza, where there was significantly more data available at the time of this study. And then there was information about COVID-19. And you can see here that for either pre-COVID-19 flu or for COVID-19, there was a significant decrease in the number of steps overall for individuals who were infected precisely after the date of infection, and that's where this y-axis crosses here. And what's interesting to see about this is that, we see that for the COVID-19 individuals that we don't have as rapid of a recovery as we might see with the traditional influenza, which fits with what we have learned about the recovery rates of COVID-19 versus more traditional influenzas. And then we also see some increases in resting heart rate that are really quite clear with pre-COVID-19 flu, and some sort of similar trends that seem to potentially exist with COVID-19. And then we also have some measurements for COVID-19 that exists after, influenza that exists after COVID-19. And so what this tells us here is that we have the ability on a large scale to take a view of overall health and understand what's going on a population level if many people are wearing these digital devices. So this is made possible because so many people have digital devices. We can measure step counts not only from wearables, but also from mobile devices. So even if we are just measuring information about people's movement, we can see that based on that previous graph, that movement changes when people get infected. And then we also have other types of digital health data, including online traces from social media platforms like Twitter, Facebook, and these have been used for disease surveillance. So if this is of interest, I definitely encourage you to check out these references here. And then finally, we also have information from environmental sensor networks that can capture contextual health-related information. So this is most commonly thought about in terms of asthma and air quality, and there are lots of technologies that exist today to help us to better understand how air quality is related to incidence of asthma. So with that, the ability to pull data from wearable devices is slightly complicated. And so I think that this is a really great overview here of how data actually gets collected by these different types of digital devices. We have different types of measurements, like we've discussed, these body monitoring. So we have some physiologic measurements like movement, heart rate, temperature, and then we also have some biochemical measurements like blood glucose that tell us what's going on in underneath the skin, essentially. And then we also have environmental and location monitoring that give us that contextual information. So from there, we have a variety of different methods of logging that data and where that data gets stored, how that data gets pre-processed. And I think that this is a critical area that's worth thinking about and that we'll dive into in more detail in this talk is what's going on behind the scenes here and how can that affect our interpretation of the data that we get access to. Also important to think about is the communication of the data. Because this is a large amount of data depending on the sampling rate of the sensors that we have, it's not always possible to push that raw data over our wireless connections. And because of that, there ends up often being some tricks that are used, for example, data compression techniques, and also often some privacy and security techniques for data encryption that cause, again, the data to be slightly changed. And in all of these steps, it's again really important for us to think about how that affects our interpretation of the final data. And then finally, once the data is received by whoever it is that plans on analyzing it, there are other considerations to take into account. So sometimes data is directly pushed to company servers. So for example, if we're talking about a Fitbit watch, the Fitbit data in its most raw and accessible form gets pushed to Fitbit servers. And then that can be accessed differently by different researchers or by the company themselves. And so finally, the piece that we care about most here, which is the implementation, is how we go from this process here of the technology development and the data collection to implementation in healthcare. So how can we enable healthcare professionals to not only be able to access that data, but to be able to use it to make actionable decisions? So for example, how can it be used to communicate with patients in a way that makes sense to both the clinician and the patient? How can it be used to diagnose and treat illness? So when we talk about digital biomarkers, this is going to be a big focus of that. And then for patients themselves, what does it mean for them to be tracking data? When changes are recorded and observed, how can they decide whether this is due to inherent biologic variability, sort of natural day-to-day variance in our physiology, or whether this really indicates that something is wrong and they may need medical care? And how can we capitalize on this to facilitate early detection so that we can actually intervene when it will be most useful? So ultimately, this is the goal that we're trying to get to, is to go from this digital data to implementation. And with that, there are several challenges along the way, and a few of those I've mentioned, but I do want to bring up this project called the Open BDP that we've been leading alongside a team at UCSF that is developing the Open Health platform. And with this, our goal is to make it easier to access data from digital devices through APIs or application programming interfaces that enable us to go in and to collect the rawest form of the data, so the data at the sample level whenever possible, and to be able to convert that data into a standardized form that can be readily interpreted by digital biomarker algorithms and converted into actionable insights. So we'll hear more today about the digital biomarker discovery pipeline and OpenM Health and how the combination of these two is making this entire process here much more feasible. So now that we've learned about the importance of wearables data and how it can be used for a variety of different purposes, one might be wondering how they might start up a wearable devices study. And the first piece of starting up a study like this is to understand what are the different types of wearables data and how you can capture these different data types. So there are many different physiologic phenomena that can be captured from wearable devices. On the screen here, I'm just showing a snapshot of some of the different physiologic categories that we might capture using different types of wearable devices. So these range from devices that can be worn on the head, or even devices that can be worn elsewhere on the body but are able to capture neurology, mental health, or sleep. We can capture information about the pulmonary system, the cardiovascular system, the GI system, the metabolic system, an overall movement, and then there are also some devices that are particularly useful in women's health. So a lot of times one of the major questions that we get is, because there are so many wearable devices on the market, how can I know that the device that I want to use is appropriate for the purpose that I intend to use it for? And so I want to refer you to two different resources that are quite useful in this space. First, for just capturing the range of wearable devices that are out there. So the Scripps Research Institute has this digital health library, which essentially is a crowdsourced collection of different wearables technologies that can be applied to a variety of different purposes. And then there's also this group called the Electra Labs that also has a very similar sort of atlas of digital health technologies. So if you're not sure where to start, I would recommend going to these two resources and taking a look at all of the different wearables that are there and are available for use. So one thing that's important to consider when you're looking at different devices is the way that the devices actually work. So how do you get the measurement that is coming off of that device? So for example, if we think about an accelerometer, what an accelerometer is really measuring is physical motion. And the way that this works is it transduces a physical signal, like physical motion, through an electrical sensor that then gets converted from an analog to a digital signal. And through this process, there's a lot of signal processing that goes on for a variety of different reasons. One of the major reasons is that the amount of data and the amount of storage that's required to capture this primary or raw signal is really high. And so there's a lot of onboard processing, which means processing that happens on the device itself, that compresses those signals, reduces noise, and makes the signal cleaner and more usable and easier to transmit. So then these signals are often transmitted to a mobile device, or in the case of the Apple Watch 4 and above, it actually gets transferred directly through a wireless network to the cloud. And so one thing that's very important to realize is that in each of these steps where a signal is changed from its original form, there's something that goes on that alters the data. So it's important to understand what is it that's going on under the hood that might make you expect to see some artifacts or some problems or maybe some cleaner data. It's important to realize that what you're getting is not that primary physical motion signal, but rather a very processed signal that then ends up on the cloud. Similarly, if we're thinking about how heart rate is measured from a wearable device, we have a method called photoplethysmography, which is kind of the most common method from a consumer wearable device. And if you own or have seen an Apple Watch, Fitbit, Garmin device, sort of any of the most common commercial wearable devices, you've probably seen the green light on the back of that device. And what that does actually is measure changes in blood volume underneath the surface of the skin. So this is really a proxy for heart rate, because we know that with every beat of the heart, the volume of blood changes underneath the surface of the skin. But again, it's important to realize that there's actually it's a proxy measurement for heart rate. So we're not actually measuring here the electrical beats of the heart or any mechanical beats of the heart, but rather we're measuring a downstream phenomenon from a heartbeat. So what that might mean is that there are other factors that can play a role in how this signal is measured. For example, blood perfusion under the skin plays a role. So if somebody is very cold, it can be difficult to get a clean PPG signal. So these are just key things to remember when you're working with any sort of wearables data, is what's really going on under the hood. And so in this case, the way that PPG works is it emits a light, and the light is passed through the skin, and then the reflected light is then detected. And the absorption of this light wavelength changes based on that blood volume underneath the skin. So you can imagine that there are a lot of different factors that can be at play here. And so in my lab, what we're doing is looking at a variety of different topics that can relate to getting artifacts or a decrease in the signal-to-noise ratio in optical heart rate monitoring, which is that PPG heart rate monitoring. So one of the areas that we've worked pretty closely in is the detection across diverse skin types. And I will mention in a moment the study that we did to analyze how well these different devices work for different people under different circumstances. Because one thing that's key to remember is that a measurement is not a measurement is not a measurement, which means that the circumstances of use can affect the measurement that we get. So if you're expecting to see that when somebody is sleeping at night versus running on a treadmill, you can equally trust the heart rate measurements from both of those circumstances, that's actually not true. And so we'll see an example of that in a moment. We're also speaking again to this idea of different sorts of activities relating to different accuracies in heart rate. We're looking at optical noise and motion artifacts, which is kind of a very well-known issue in optical heart rate monitoring. We're also looking at this crossover problem, which is the mixing of periodic signals. Because that onboard processing that I mentioned before often is designed to detect a periodic signal, because we expect that the heartbeat is a periodic signal. Sometimes it can actually be tricky because when we're walking, we actually generate a periodic signal from the steps. And so what can happen is the algorithms that are aimed to clean that optical signal and search for a periodicity can often lock onto the wrong periodic signal. So we'll actually lock onto, for example, the step rate rather than the heart rate. So this is something that's important to be aware of. And then finally, the measurement location on the body really matters. So you can have a PPG sensor that sits on the finger, which is something that we commonly see in hospitals, and often we'll see that that actually is a different wavelength of light, and we'll talk about that in a bit. But we can also have measurements from the wrist, we can have measurements from the chest, we can have measurements from the earlobe. So there are a variety of different measurement locations, and each of those come with some benefits and some challenges. And so it's important to, again, realize the context of the use in order to be able to interpret the data most appropriately. So ultimately, what my lab is working on is developing methods to jointly tune the hardware and the software across all body types and circumstances of use, so that we can be confident that when we're getting a measurement of heart rate from a PPG sensor, that this is actually measuring heart rate. And if we were to compare it to a gold standard of electrocardiography or echocardiography, that we would see that the beats that are represented on the PPG signal are the same as the beats that are represented in the gold standard. And so the two things that I wanted to come back to and mention were this issue of detection across diverse skin types and looking at optical noise and motion artifacts. So one of my very talented PhD students, Brene Bent, who you can find here on Twitter at runsdata, worked with me on this project to look at how wearable devices, PPG sensors, capture heart rate accurately across a variety of different skin tones and a variety of different activities. And the reason that we were particularly interested in skin tone is because there was a lot of anecdotal evidence and also a very clear engineering theory that darker skin would actually have less accurate heart rate measurements. And the reason for that is because darker skin has more melanin content, and melanin actually absorbs more of that green wavelength of light. So the idea is that it would be harder to get the reflected light to detect these changes in blood volume under the skin in people who have more melanin content. So that would mean people with darker skin. And so we performed a very detailed analysis to probe whether this was the case in a variety of different devices. So we used consumer wearables, which are things that most people would buy off the shelves. We looked at the Apple Watch, Fitbit, Garmin, and a watch that's very popular in China, the Xiaomi Mi Band. And then we also looked at two devices that are more commonly used in research, the Empatica E4 and the BioVotion device. And interestingly, two of these devices, the BioVotion and the Garmin, actually had a disclaimer on their website saying that these measurements might actually be less accurate in people with darker skin tones. So that was even more interesting to us to see if that was in fact the case. And so what we found was that across the different skin tones, we actually didn't see a change in accuracy as we had expected. And I think this is one of the reasons why this type of study is so important, is that there was a lot of anecdotal evidence, and I think that this probably motivated companies to really work on their detection algorithms and the way that they record PPG signals. So we found that actually the light that is passed through the skin is of a higher intensity in people with darker skin tones. So there's actually a built-in mechanism to a lot of these devices to account for the fact that somebody has darker skin. But what was also interesting about this is that because we also tested this across a variety of different activities, we found that actually the average accuracy of heart rate measurements was decreased by about 30% when somebody went from rest to walking at a moderate pace on a treadmill. And so when we think about the times when we might be using these devices, for example, maybe to monitor someone's health during physical activity, it's important to know that the PPG-based heart rate measurements are actually not very accurate. And so there's a lot of work to be done in this space. And one other thing that I'll note on the subject of skin tone that's become it's become more top of mind these days with the COVID-19 crisis, is that blood oxygen monitoring uses the same sort of technology. So blood oxygen monitors are optical-based technologies. Usually it's a clip on the finger or the ear, and we see this commonly used in hospitals. And I had mentioned that I would talk about the difference in wavelength of light. So typically these devices actually use red light rather than green light, because red light is affected more by a motion artifact, but in a hospital setting, people are normally laying very still. And so the reason why that's important is that the red light is supposed to be a better wavelength for capturing these changes in blood volume or changes in hemoglobin content, oxygenated hemoglobin content underneath the skin across different skin tones. But a paper came out actually last week showing that for people with darker skin tones, the coherence between blood oxygen measurements from this optical-based sensing in the hospital as compared to the arterial blood gas, which is a much more invasive technique, shows that there's actually a very big difference only for people with darker skin. So what was shocking about this is that the measurements being taken for people with darker skin are actually potentially missing people who have very, very low blood oxygenation that aren't being caught inside the hospital as needing intervention, even when they do need intervention. So again, this comes back to why it's so important in engineering and in data analysis that we think about the circumstances of use and that we think about the technologies and the way that the technologies actually capture the physiology. Because otherwise we might take the data at face value and miss some important points about the way that the data tells us what the physiology is. So, some of the biggest benefits of wearables are that they bring the ability to monitor health outside of the clinic. And so, often when we think about vital signs measurements, we think about measurements that are taken at the beginning of a typical clinic visit. But what we can consider is that if we have accurate measurements coming from these devices, we can actually get continuous vital signs measurements. So, not just at a single point in time, but rather every 5 minutes or every minute over the course of someone's lifetime, not only in a hospital setting, but in somebody's home, in remote or rural regions, and in low resource settings. Because the underlying hardware for these devices is actually very cheap. So if we think about it, the LEDs and the hardware encasings together cost, under roughly $5 to $10. And there are devices in China like the Xiaomi Mi Band that sell for $15 to $20. So we can envision how, as these technologies get better and as the manufacturing becomes more reliable, we'll end up seeing these devices in use across the world in all sorts of settings. And one of the biggest challenges actually to using these devices for large-scale healthcare is that there's this process of the data collection and transfer that relies on a reliable network connection. So whether that is a connection to a wireless network or whether that's a hardwired connection to the internet, it's important to understand that the limitations of getting the data from these devices actually have to do with the way that the data is transferred. And so one of the things that we're working on these days is data compression methods to better enable local storage for a longer period of time in locations where there might be unreliable connections to dump the data onto the cloud. And then in other cases, enabling an easier transfer of that data. So the smaller the data that needs to be transferred, the easier that transfer is. And then ideally, what we hope to see is that there's an automated method that links up these wearable devices to some sort of actionable health conclusion. And so whether that is engaging healthcare practitioners or telehealth, or just some automated adaptive just-in-time interventions using AI-based technology. I imagine that we will see someday that this will be a closed-loop system where the measurements will directly result in some action. And that can be in a variety of different health areas. So we might see, and we're already actually seeing in the cardiovascular space, for example, with AFib detection, that an Apple Watch can directly tell you if you are at a high risk of having AFib events. And so there are a lot of challenges, and I definitely don't want to gloss over this point that the current environment for these wearables is such that there is a lot of potential, but a lot that we need to address. We've talked today a little bit about accuracy. We have not touched upon at all privacy and security issues, which are sort of a whole field in and of itself that many people are working on. I'll also mention with oversight and lack of standards, this is an area that we've been getting into in more detail because it is very tricky to understand how well we can trust the data coming off of these devices, given that they are not products that are regulated by the FDA. And so I would really encourage you, if you're interested in better understanding what the regulatory status is of these different devices, to check out the Digital Medicine Society. We're doing quite a bit of work with them in trying to better establish standards, and there is a resource called the Playbook through the Digital Medicine Society, which helps to give sort of an overview of if you're using wearable devices or wearable devices data, maybe in clinical trials or in actual care settings, how you would use that data appropriately. So we talk a lot there about fit for purpose use. And again, there are various other challenges that will need to be overcome, but I think as we're thinking about what the possibilities are for these technologies, it's clear that it's worth our time to figure out how to overcome these challenges and limitations to make sure that we can develop equitable technologies that bring healthcare to many more people than once had access, and also bring more continuous and personalized healthcare to all. So now that we understand some of the nuances of wearable sensors and where some of the challenges can come in with data accuracy, we may want to know how we can actually use wearable devices' data toward clinically actionable decision making. And so with that, we can introduce this concept of a digital biomarker. A digital biomarker is digitally collected data, so for example, heart rate from a vital sign monitor, that is transformed into an indicator of a health outcome, for example, prediabetes. And so the idea here is that we're taking data that comes from a wearable sensor, and we are discovering a mathematical relationship to some indicator of a health outcome. And so maybe that is a clinical measurement like blood glucose that is usually used to indicate diabetes or prediabetes. And so this concept is not that different from a lot of the current uses of wearable devices for clinical decision making. So there are kind of two major categories that right now are very useful for wearable devices data. One of those is in detecting infectious disease, and the other one is in detecting chronic disease. So for the acute infectious disease, typically the combination of heart rate and skin temperature, and sometimes also pulse oximetry, give us information about whether somebody's physiology is normal for them or whether it has changed abruptly, likely due to an infection. And on the chronic disease side, we've seen that there are measurements of resting heart rate, physical activity, and sleep that can help to indicate whether or not somebody has a chronic disease. And there are lots of different methods to relate the data from the devices to the outcome. And so you can imagine that, much like a lot of the more traditional statistical learning that we do, or machine learning that we do, we can determine what the likeliest relationship is and then build models based off of that. And so in order to be able to do this accurately, we need to have faith in the technologies that we're using. So we've been working closely with the Digital Medicine Society to develop language around digital medicine tools that help us to better understand their accuracy and how we can evaluate them. So the first, vocabulary word that I would want to introduce here is a biometric monitoring technology. So because there's been this rapid rise in the demand for and development of digital medicine products, we've seen that there's not been the supporting body of evidence that tells us about their quality, safety, and efficacy. Because these devices are not FDA regulated, we end up with a lot of tools that we don't fully understand. And so we don't want the data from these tools to be misinterpreted. And particularly as we've seen in the context of clinical trials, more and more connected digital medicine tools being used, we want to make sure that data is not misinterpreted in a way that leads to misleading study conclusions and possibly to patient harm. And so here what we've done is we've developed this lexicon for talking about wearables in the context of digital medicine. So a biomet, or a biometric monitoring technology, is a connected digital medicine tool that processes data captured by mobile sensors using algorithms to generate functional measures of behavior and or physiology. And so what we can do is we can then take this concept of a digital biomarker and relate it back to these tools that are collecting data, and then from that data making some sort of a conclusion about the physiology or behavior. building on this idea, we've looked at existing frameworks for new biotechnologies and discovered that in many cases they were not sufficiently adaptable, but they did provide meaningful insight for developing new evaluation frameworks for biomeds. And adopting streamlined methods for transparent evaluation processes coupled with transparencies, this will help us to overcome the black box technology development that has caused biomeds to be used in ways that are often not fit for purpose. So how do we best evaluate biomeds and digital biomarkers? Well, first we need to think about what the term clinically validated means, because it's important to recall that biomeds are not regulated by the FDA. And so if a device or a company is claiming that something is clinically validated, that can mean a variety of different things. So it's important to dig in and really understand what is meant by that phrase. And again, because there is no systematic or evidence-based evaluation framework, we've worked closely with the DIME Society, the Digital Medicine Society, to come up with a method that allows us to understand the accuracy and the validity of measurements from these biomeds. And so through that, we have a 3-stage process that we've developed from verification, which is almost like the bench verification, making sure that the tool works as intended. And the way we think about this is if we have an accelerometer and we put it on a shake plate where the RPM are well established, that we are able to replicate that in the accelerometer. And then we have 2 validation processes. Both of those are, if we are to take the biomet and put it on a human, do we get, 1, for analytic validation, measurements that we expect in terms of physiologic phenomenon? So if an accelerometer purports to measure a step count, do we get the same step count as would be measured by a person watching somebody walking and counting their steps? And for clinical validation, if we expect that there should be a relationship between steps or between movement and a disease like MS, for example, in clinical validation, we're looking to see that we see that relationship between our accelerometer and the disease state. And so some of the things that can make it tricky to perform this verification and validation process is that we have these breaks in the data supply chain. And so at the point where we actually access the data can matter. And so it's important for us to define that data supply chain as the data flow and data provenance so that we understand what's been manipulated in the data and at what points in the data supply chain, where information goes from hardware, sensors, software to algorithms. So to dive into a bit more detail on what we mean by verification, analytic validation, and clinical validation, we can look to this chart, and I would also refer you to this paper that we put together as a team in the Digital Medicine Society to better explain the different methods for evaluating biomeds. And to clarify what exactly these different steps entail, if we're looking at verification, what we're doing here is evaluating and demonstrating the performance of a sensor technology within a biomed and the sample level data it generates against a pre-specified set of criteria. With analytic validation, we're evaluating the performance of the algorithm and the ability of this component of the biomet to measure, detect, or predict physiological or behavioral metrics. And then finally, with clinical validation, we're evaluating whether a biomet acceptably identifies, measures, or predicts a meaningful clinical, biological, physical, or functional state or experience in the stated context of use. And in parentheses we have here, which includes a specified population. And that's very important when we think back to the original challenges with ensuring that devices are measuring what they say they're measuring in different skin tones and in different activity types. And so one thing that's important to note also about this is that this process is iterative. So when we're using wearable devices, oftentimes something that's not really thought about is the fact that through these through all of these steps, there's a lot of software or firmware that's involved. And so if you're using a Fitbit version 2 and you haven't changed the hardware and everybody in your study is using the same version of the device, but actually the company pushes a software update that changes the way a heartbeat is detected from PPG, for example, and if you're not aware of that change in the firmware, then actually your data can change at a certain point in the study without you knowing. And so it's really important to be aware that this can happen, that even if you believe that you're using the same device over time, that actually may not be the case. That changes to software or firmware or the way that data flows or the way that data is compressed can change the integrity of the data. So whenever there are changes to hardware or firmware or software or using the device in a population that it was not originally tested in or under circumstances of use that it was not originally tested in, then really these processes need to be redone to make sure that the measurements are what we expect them to be. So then the goal is that we want to make sure that we're extracting useful information from the wearables data. So really that's that third step there, the clinical validation, to say if we have this information from wearables data and we can trust it and we know that it measures the physical or behavioral phenomenon that we expect, how do we know that that information is useful, and then how do we apply it in real life? And so that's where a lot of this biomedical and health data science work comes in, where our goal is to extract useful information and knowledge from large volumes of biomedical and health data. So in this case, we're talking specifically data coming off of wearable devices in order to improve medical decision-making or providing health insights to make data-driven medical decisions. So what we're trying to do is tie that wearable device's data to a health or medical decision. And again, we've talked a little bit about some of the potential issues with data, but when we get the data from the devices, there's still quite a bit to do. So there's a lot that needs to be done in sort of the pre-processing range, making sure that we are understanding when there are missing values in our data, what those can be attributed to. And so we know that there are many different types of missingness, and it can be important to understand in the context of the device itself, are these data missing at random, missing not at random, and if so, how should they be handled? For example, are there people who consistently don't wear the device when they're sleeping? And if so, if you are developing a digital biomarker that is mainly driven by measurements during sleep, then are you even able to use the data from that patient or that study participant? We're also looking for range consistency. So there's a lot of domain knowledge that can get injected into this process of understanding what is a reasonable value for heart rate, for example. So if we see measurements of 0 or measurements of 500, we know that likely there's some issue going on with the data. But then the question becomes how to handle that. So do we not use any of the data from that person or from that device? Do we transform that data in some way? And that can be a really use case specific question. We also want to work on denoising. So like we said, there are some circumstances of use where we know the data is less reliable. For example, during high levels of physical activity. So there are a lot of options there of completely removing data that was measured during a certain level of physical activity, and that can be defined based on the accelerometer or based on the step count, depending on what level of information you get. And then finally, before we are going to develop any sort of a prediction algorithm or any sort of explore any sort of relationship between our digital data and the clinical outcome of interest, we want to make sure that the methods that we're using are appropriate for the data that we have. And so I think that this is an audience that I probably don't need to emphasize that point too much for, but again, there are very different shapes to the data that we see here. So for example, we typically see somewhat of a normal distribution for heart rate, And skin temperature can be a little bit skewed, but for the most part, a pretty normal distribution. But when we're looking at some measurements like galvanic skin response or electrodermal activity, or something like accelerometer, what we see is that these are incredibly tailed distributions. And the reason for that is because the very high measurements for at least these two parameters occur rarely. So the accelerometer, we may get only the highest measurements when somebody is in the process of running, for example, which may only happen one time per week. And if we have one measurement per second, you can imagine how that really skews the data. And so it's important to think about how the different sensors that we have result in different distributions of the data. And then ultimately making sure that the way that you are handling the data is appropriate for whatever prediction or relationship you're trying to uncover. So when we're trying to develop these relationships, we're really focused, at least in my lab, on using statistical learning. So we want to understand how multiple different variables, usually measurements from wearable devices combined with some demographic covariates, work together to affect some outcome. So really basically, we'll ask, how would a drug A affect tumor progression and our demographics associated with its efficacy. So in this case, we might say, how does a digital intervention to increase physical activity affect the physical activity? And then one step further, is that related to some health outcome? We might also look at a combination of other measurements, like patient-reported outcomes. So for example, we could look at how diet, which would be a patient-reported outcome, and physical activity, which could be measured automatically from the device, affect weight. So our goal is to develop an accurate model that can be used to predict some outcome Y on the basis of these variables from the digital data X. In order to understand which method is most appropriate, we want to think about whether our outcome is quantitative or qualitative, and whether the information that we're getting from the device itself is quantitative or qualitative. So we might see that we're trying to predict a step count, for example, from accelerometry, which would be more of a regression problem, or if we want to predict sleep stages, then that's really more of a classification problem. And so, as we know, some methods can either use quantitative or qualitative data or both. And so we want to be really clear about what the purpose is of the use of this data and what it is that we're trying to predict. So then the question may be that we want to evaluate how good a digital biomarker is. And so there's a couple of ways to think about this. And one way we can think about this in the classical sense of evaluating an algorithm. And if we're looking at regression, typically we're looking at, for example, MSE or R squared. If we're looking at classification, we may be looking at accuracy, AUC, F1 score, and so on. And if we're looking at clustering, we may look at sort of how much information we're able to explain through this clustering process, so maybe AIC and BIC. But we can take this even a step further to say that we have a measure of how good the algorithm itself is, but then we want to take that again into the broader context of this clinical validation question of, even if the algorithm on its surface is quite good, if we were to extend this to a new population, how does this perform? If we were to extend this to a slightly different use case, how well does this digital biomarker perform? So keeping in mind the purpose is always really important when we're trying to determine what it means for a digital biomarker to be good or good enough. So with that, we're going to switch gears and talk about some examples of some research topics where we've been using wearable device data to predict some sort of health outcome or to better understand health status. I'll start by talking about one of the studies that I worked on as a postdoctoral fellow at Stanford with Mike Snyder. And this study is the IPOP study, or Integrative Personal Omics Profiling. And the idea here was to integrate a variety of different data types from multi-omics to wearable device data and clinical lab tests for personalized type 2 diabetes risk assessment and precision therapy. And the way that this study looked was that we had roughly 100 individuals that were pre-diabetic and healthy that were monitored over the course of several years. I think now it's roughly up to 6 or 7 years. And throughout this time, people came into the clinic 4 times per year to give samples, and those samples would be used for multi-omics testing as well as more traditional lab testing. And so these were considered to be healthy visits where we're capturing lots of biomolecular information about a person. And during this entire study, people were also wearing wearable devices that were capturing measurements of heart rate and accelerometry and sort of other physiologic parameters over time. And so what's really interesting about this sort of a study, a longitudinal study, is that there are natural perturbations that take place. So people would get infections or have stressful life events or undergo some dietary change. And during that process, we would ask people to come in to give more dense samples so that we would actually get a very high resolution picture of what the biomolecular changes look like over time. And so with this data, I'm only focusing in this talk on what we did with the wearable devices data and how we related it back to some of these health outcomes. But there's a large range of studies that have come out of this initial IPOP study that focus in on the biomolecular changes. And so I would really encourage you, if you're interested, to take a look at several papers in this area on the IPOP study so that you can see more about the biomolecules as well. But in the digital health study, this was a paper that we published in 2017 in PLOS Bio that is really focused on how we can use data from wearable devices in a way that helps us to understand physiology and potentially detect certain important things about health. So we had tested a variety of different devices, and as I had mentioned at the beginning of this talk, there are a lot of different devices to choose from, and it can sometimes be tricky to understand which devices are most relevant for what you're trying to measure, particularly in an exploratory study of this kind where we're not actually sure what exactly it is that we can learn from these different types of measurements. And so we tested a lot of different devices. We actually had explored, I think, roughly 500 devices for the usefulness for this study, and things that we analyzed in that process were the usability of the device, because if we have a device but people don't use it, then it's really not all that useful. And we also tested for the accuracy of the data. So was there evidence out there that this data would replicate some clinical gold standard measurements? We also checked for how easy it was to actually access the data, because this turns out not to be a trivial question, is that there are a lot of devices out there, but it can often be very difficult to get the data from the device or from the device company. And so these were kind of the three major considerations that we had when we were evaluating devices for use in this study. And then ultimately, the analyses that I'm going to focus in on today will be using the Basis Watch, which was a device by Intel that measured 4 different parameters, and I'll talk about them in a second. And we could use them to do some overall analyses on physiology and activity, and then we could also relate them back to some clinical outcomes, from insulin sensitivity to inflammation. So to take a look at this, actually I'm only focusing in on 3 out of the 4 measurements from this watch, but we had heart rate, and again that's using the optical heart rate monitoring that we talked about earlier, using the green light on the back of the watch. We have skin temperature, and we have accelerometry. And what we can see here on the x-axis, each of these ticks is a different person in the study, and looking at the blue triangles, these are average measurements of skin temperature, and the red circles are average heart rate, and then the black is the number of days monitored. And so what's pretty interesting at first glance about this data is that we can see that skin temperature has a pretty tight range. And this makes a lot of sense when we think about the context of temperature, that If temperature varies too much, then we're actually in a very dangerous physiologic state. But our heart rate has a much wider dynamic range. Something that's even more interesting to think about, though, that I like to consider is that there's actually a big difference also in the dynamic range within an individual for heart rate. And so what we can see here is that we might have two people that have very similar heart rate values. So like this person here compared to this person here. That if we were to just look at their mean heart rate, we would say that these two people have very similar mean heart rates. However, if we were to look at any single measurement of heart rate, if we had a measurement that was maybe up here, we would say, okay, this is in range for this person. This falls within 1 or 2 standard deviations from the mean, and so we expect that this would be, maybe a little bit out of typical range for this person, but nothing to be too alarmed about. Whereas if we saw a value up here for this person, we might actually be quite alarmed about that because this does not look typical for this person. And so it's really important for us to consider when we're building these digital biomarker algorithms that are detecting physiology that might be indicative of disease to really understand how that relates back to somebody's own personal context. And so we can also see this in some circadian biology. So, for example, we know that typically during the night, the heart rate decreases during sleep, and we can see that for most people in our study here. However, you can see that there are some people for which that dip in heart rate is much more prominent versus maybe a little bit less obvious. And then we even have some people where actually the opposite is true, where their heart rate actually increases at night. And it's interesting to think about because if our goal is to detect abnormal physiology, then we might expect that, anybody who has a heart rate increase at night might actually be abnormal. But when we look at a person within their own individual data, that actually might be normal for that person. And the same thing goes with a change in skin temperature. So again, we see that skin temperature tends to increase at night. And again, we see that for almost everybody in our study, but not for every single person. And that change happens to different extents. And what's also really quite interesting is that the people who have the more characteristic heart rate changes, we might expect to have the more characteristic skin temperature changes, and vice versa, the people who don't have the most characteristic heart rate changes not to have those characteristic skin temperature changes, but that tends not to be the case. So really the take-home message from this is that physiology really differs by individual, and if we want to build a digital biomarker that is useful for detecting changes in physiology, we need to take into account these personal baselines. Rather than just using summary statistics, we can also use the shape of the data in terms of the overall circadian activities, because we have this ability to take a look at the longitudinal information. So what I'm showing here is taking the average circadian patterns for different individuals. And so what you can see here is essentially this is based on the accelerometer. So this is just movement over the course of the day. And on the x-axis here, we're looking at the hour of the day. So we've aligned people based on the time that they typically sleep, and we use this alignment period as a combination of the accelerometer and heart rate, which is used to indicate sleep. And what we can see is that you can see that there are dramatically different kind of categories of activity, where we have some people that are more active earlier in the morning, like people with this first peak. And then we have some people that have certain activity peaks throughout the day. So people in this lower left-hand quadrant tend to have 2 peaks in activity throughout the day. We saw that there were some people who have 3 peaks. And actually these 2 peaks seem to correspond more to active commute times. And because these were primarily individuals measured in the Bay Area, there are lots of active commuters there. And this made a whole lot of sense in terms of commute times. And then we also saw that these 3 peaks tend to correspond with meal times. And so these may be individuals who go out to breakfast, lunch, and dinner, for example. But what we could do here is, based on the shape of this daily curve, we could perform functional clustering. And so this would it's an unsupervised method to group people together based on the similarity of their daily activity habits. And so this is just one of many things that we can do with wearable device data. And then finally, we can take a look at some of the relationships between the different measurements that we get from the smartwatch. So here what we have is the measurement of heart rate based on the activity. And so as we might expect, if you're standing on a treadmill, as you start to increase the pace of the treadmill, your heart rate starts to increase. And so that was kind of the expectation with this analysis. And what we saw was that, in fact, there is a pretty monotonically increasing relationship for almost everybody in this study between their heart rate and their activity. And so what's really interesting about this, though, is if we actually look at the correlation coefficients between activity and heart rate, we see that for most people, the two are very tightly correlated. But there are a few people for whom that's not necessarily true. And so what we've started to do is dig into these few individuals in this study to try to better understand whether there's something about this relationship between heart rate and activity within an individual that tells us more about their cardiovascular fitness or their overall health. And so I think one of the most exciting things to come out of this study was the ability to actually detect illness from the devices, because everything I've showed you up until this point is sort of some more aggregate statistics and some overall observations about the smartwatch data. But really what we wanted to do was relate this back to health measurements. And so this was an algorithm that I designed called the change of heart, which was essentially a method to take every measurement of heart rate or physical activity or skin temperature or galvanic skin response and compare that to your personal average. And that personal average could be defined in a variety of different ways. And so essentially what we could do is, because each of these sensors were taking one measurement per second over the course of the day, so that's 86,400 measurements per sensor per day, we could take every one of those measurements and compare it to some sort of a mean. So we could define that mean based on the entire monitoring period. We could define that mean based on the same time of day over the course of the monitoring period. We could do weekdays versus weekends. We could do time of year. So there are a lot of different ways to define this mean. And I think that when we talk about fit for purpose, that definition is really critical. And then we could bin the measurement. So we could say that this measurement is more than 2 standard deviations from the mean. So that would fall into kind of this bin of being uncharacteristically high, or we might have it falling in a bin where it's uncharacteristically low. But for the most part, we expect that most of the measurements fall within 1 standard deviation of the mean and would be considered normal for this individual. So taking an example of one person where we applied this method in our study, you can see that this is about a year and a half's worth of monitoring on the x-axis. And you can see that, there are times when there are missingness because maybe the watch broke or the person didn't wear the watch for that period of time. But what's really interesting is that we see that looking at these bins for the heart rate and then these bins for the skin temperature, that there are some blips on the radar that really stand out. So I think this area here is one of the most obvious. And when we looked back to the clinical records for this person, we found that that blip on the radar corresponded to a Lyme's disease infection, and that we could, in fact, validate that this was a date where we had an infection and that it aligned perfectly with this blip on the radar from the wearable devices data, and that we had the positive antibody screen for Lyme's disease. So again, I want to emphasize that this method was not specific to detecting Lyme's disease, but rather was able to detect an abnormal period of physiology that we could then use to go back and try to learn what happened in this cohort. And so for actually this same person, what we did was we went back to their clinical records, and recall the study design here is that people were coming into the clinic for both healthy visits and sick visits. So they were coming in a minimum of 4 times per year to give samples for what would be considered healthy visits, and then they would also come in more often when they were feeling sick. And so plotting here the proportion of skin temperature measurements that are personal outliers, so based on the number of standard deviations from the mean, and then the percent of heart rate measurements that are personal outliers, we can see that there are some days here that are kind of most abnormal for this person. And those most outlying days correspond to that blip on the radar, that Lyme disease infection that we just saw before. And so there were 5 days where this person came into the clinic during their Lyme disease infection, and we saw that for all of those 5 days, their smartwatch data looked quite abnormal. But what's interesting is that there are also other outlying days. And so we looked back at the clinical records and we found that one of those corresponded to a viral infection. And then there were 2 other dates that didn't have anything noted by the individual themselves. They weren't reporting feeling sick. But in fact, in their what was supposed to be a healthy measurement, we actually saw that there were high levels of a protein called C-reactive protein, which is indicative of infection. And so what's really interesting here is that this person was actually at a high level of inflammation, even though they themselves didn't know it. And so the implications of this are really interesting because when we think about asymptomatic infection or pre-symptomatic infection, this gives us an opportunity to be able to detect subclinical infections maybe before they would become infectious or before somebody could spread a virus. And so the idea here now is that we want to test this in a much larger population. So in the study itself, we only had about 9 people who actually naturally got sick over that time period. And so we were able to test this algorithm in those few people, but it was really not sufficient to be able to assess on a much broader level the sensitivity and specificity. But the learnings from this study were essentially that we can use these individual baselines to develop digital biomarkers that enable us to identify acute or chronic illness early. And so we can extend this concept, this use case, to a variety of other use cases. So we're working in a lot of areas now with wearable devices to see if we can develop digital biomarkers to detect or predict circadian disruption, working with shift workers who sometimes sleep during the day and sometimes at night. We're looking to detect health improvements from sleep and exercise. We're looking to understand how contagious a person is if they are sick. So if they have a virus, how much virus are they shedding? The susceptibility to contracting illness. So we have a study in collaboration with the Duke Center for Applied Genomics and Precision Medicine where people actually underwent flu and rhinovirus challenge studies where they were exposed to the viruses. And surprisingly, some people don't actually contract the virus even when they're exposed, and we don't fully understand why that is, but digital biomarkers can help us to better understand what makes somebody more susceptible to contracting an illness. We're also looking at prediabetes prior to symptoms, so to see if we can detect prediabetes before somebody might know that they're sick. And then we also have some work in women's health space, understanding the risk of preterm birth. And more concretely from the IPOP study that I was just mentioning, there was also an interesting finding that we saw a relationship between insulin resistance status or insulin sensitivity status and changes in heart rate or steps. And so what's interesting about this is that insulin sensitivity is a precursor to prediabetes and type 2 diabetes. And as somebody becomes more insulin resistant, they're moving closer and closer toward that diabetic state. And so what's well known is that physical activity plays an important role in the prevention of prediabetes and type 2 diabetes. But here, the preliminary data indicates that there's really this relationship that we can see between not only physical activity, but also resting heart rate. And so what we're working to do now is to better understand how we might tease apart this relationship and maybe use information from a non-invasive smartwatch to better predict who might be at risk for prediabetes. And to do this, we developed a study along with the late Dr. Mark Feinglass in Duke Endocrinology, and now working with Dr. Matt Crowley, where we have 16 people enrolled in a study that were pre-diabetic and high-risk normoglycemic. And every day for 2 weeks, they were wearing 2 different wearable devices. One is a device that measures the same 4 metrics that I mentioned earlier. That's PPG-based heart rate, accelerometry, skin temperature, and electrodermal activity, which is a proxy measure for sweat or for stress or emotional arousal. And then there's also a very similar device called the BioVotion, which is worn on the upper arm. And throughout this time, people also wore a continuous glucose monitor, which enables us to get one measurement every 5 minutes of somebody's interstitial glucose. And so what we could do here is find relationships, if any exist, between the glucose and measurements from the smart devices. And so every other day we had individuals eat Frosted Flakes, which induce a glycemic response that we can actually see on the continuous glucose monitor. So we know exactly what somebody ate, and this is the first thing they eat in the morning. And the reason that we do every other day is because there are some known circadian effects to glucose. And so we wanted to be able to rule those out. And so at the end of the study, we had individuals perform an oral glucose tolerance test where we get several clinical measures of somebody's response to a glucose assault. So we get to see how is their physiology changing in a very, very controlled setting. And so finally, what we're working to do with this data is to develop some early examples of digital biomarkers that maybe show a relationship between this continuous glucose monitoring data and the smartwatch data. And there's a lot of hypotheses on the underlying biology of why there would be relationships between this invasive metric and this non-invasive metric. And what we hope to do is be able to develop a technology that can be deployed on a much larger scale to detect prediabetes. Some of the early findings from this study are very much related to what we've seen in the past, which is that having a personal baseline gives us a much better ability to develop accurate models. And so what we were specifically looking to do here is develop prediction models of glucose based on measurements from the non-invasive watch. So we were aiming to predict measurements from the continuous glucose monitor using a combination of measurements from the heart rate and movement, skin temperature, and electrodermal activity. And so what we can see is that there's this crossover point where when we have information from the personalized model, but we don't have a lot of historical data on an individual, the accuracy of this prediction model is actually quite low. And it increases as time goes on, as we have more personalized information or more baseline data. And there's some point when we have population level data from other people that we can use to train the model that does quite well. But at some point, having a better personal baseline is more powerful than using population level parameters. And so what we're showing here is that there's this interesting crossover point at which once we have a sufficient amount of personalized data, that it makes more sense to build personalized models than to use these population level parameters. So in addition to detection of glucose and glycemic parameters from smartwatches, we are also working on some other areas where digital biomarkers can be quite useful. So one of those I've alluded to a couple of times is something that's commonly existing on Apple Watches, and Fitbits and other devices are also getting these AFib detection methods. But what we've been doing in my lab is working on the detection of other types of arrhythmia, so not just atrial fibrillation, but there's a variety of different kinds of arrhythmias that can exist from PPG signals. And so there's a lot of steps that are involved here, and I'm just going to very briefly mention what we've been working on. So one of the biggest challenges when we're thinking about trying to develop a digital biomarker from a measurement that is more accurate as a sort of a clinical gold standard. So the example here is the PPG from the smartwatch versus an ECG that would normally be taken in the clinic, is that we need to be able to develop a method to align those signals so that we can be sure that when we're annotating a signal for outcomes, that those are correctly annotated. And so some of the tricky things with wearables and with biomets in general is that there can be signal drift. So that if we have two signals that are measuring the same phenomenon at the same time, over time we can actually get changes in that time signal. So even if the signals align in some points, they may not align in others. And so a good example of that, if we take a look at this signal here, is that if we're looking at the black ECG and the blue PPG, is that we can see that there's some alignment here, and you lose the alignment between the ECG and PPG in certain places, but then the alignment sort of comes back. And so we developed in a dynamic time warping method to be able to align wearable signals based on specific events in the signals. And so that's helped us to be able to compare the wearable with the clinical gold standard in a way that accurately incorporates the physiology. So there are some steps that are involved here that involve the extraction of the periodic signals in the appropriate way. And so like I had mentioned, when we're talking about heart rate, we can often have crossover of different periodic signals like heart rate and step cadence. And so there's some important steps here for denoising. And then ultimately, when we talk about arrhythmia detection, what we're trying to do here is be able to detect abnormal cardiovascular events. But the tricky part is that when we're working with an ECG, we have actual electrical measurements from the heart, whereas with PPG, we're measuring changes in blood volume under the skin. So again, understanding those differences is really necessary for us to be able to determine whether there are variables from the ECG that can be adopted for use in PPG, or whether those differences in the way the measurements are taken are really going to drive differences in how the models are developed. And finally, the goals that we're moving toward with the development of digital biomarkers is to be able to automate that process. So to close the loop between the measurements that we take and the interventions that we deliver. And so the area that we are quite focused on now is with adaptive just-in-time interventions. And so we have a couple of different projects going on in my lab where we are developing a just-in-time intervention for dietary and physical activity interventions to get the right message to the right person at the right time based on their typical habits. So for example, if we know that somebody typically frequents a specific fast food restaurant at a certain time of day, then we can intervene and offer a coupon for a healthier menu item. And so what we're doing here is ideally making these digital biomarkers actionable in a systematic way. And so that is kind of the end goal with this work. So with that, I want to switch gears and talk more about some of the ongoing efforts to develop digital biomarkers of influenza-like illnesses and COVID-19, because I think this work is particularly relevant in today's world. And so this work has been going on kind of simultaneously. So I had mentioned some of the work that I did while at Stanford on the infection detection, and ongoing at Duke and with some partners from Duke at the same time was this work to develop digital biomarkers and also biomolecular biomarkers to predict infectious disease susceptibility and also to be able to detect infection early. And so this has been a collaboration with the University of Virginia, University of Michigan, Duke, and DARPA. And so what these studies look like is a viral challenge model where we have healthy volunteers that are exposed to a specific kind of virus, and then they are observed for 5 to 10 days, and we see people who get infected versus people who do not get infected. And then we also see the severity of the infection. And so this has been done with several different viruses at this point. And what we're doing is trying to compare or trying to actually predict the clinical measurements that we would normally have. So we get information based on an RT-PCR about somebody's positivity status for infection. We have subjective and objective symptoms that people report. And then our goal is to use the measurements from the smartwatch to be able to predict some of these clinical parameters. So using a similar method to what I had mentioned earlier, this functional clustering, we've actually been able to use data-driven methods to group individuals by infection severity. And so what we found was that when we encode the subjective and objective symptoms and the clinical measurements of illness, so the RT-PCR data for infection positivity status, we see that we can actually discern 3 groups. So this was a clustering method where we don't actually give the number of clusters, but the most intuitive number of clusters that come out of this is 3 clusters, which when we think about it clinically correspond to these 3 different infection severities. And so you can see here in a bit more detail what these different classes look like. So if we're looking at the viral shedding, we're looking at a total symptom score, which can be broken down by objective symptoms and subjective symptoms. So we think about objective symptoms as something that somebody external to you could witness. So these are things like vomiting, sneezing, those sorts of things. And then we have subjective symptoms, which are things like headache, stomach aches, things that are more difficult to measure or to observe objectively. And so you can see that we have these kind of 3 different classes. And this is for people who were exposed to the H1N1 virus. So there are several people here on the x-axis that we can see really were not affected and actually didn't really get infected either by the clinical measurement standard or by any sort of symptoms with the illness. And then we can see that there's a milder version and then a more moderate or severe version of the infection. And so right now what we're working on is using the smartwatch data to predict these different classes of infection. And the goal here is to be able to detect, ideally, within several hours of somebody being exposed to a virus, whether or not they will get infected, and if they do get infected, how severe will it get? So this makes a lot of sense when we think about opportunities to curb the spread of COVID-19. So there are lots of ways that we can use wearable devices to help us try to decrease the amount of spread of COVID-19. So we have a few options in terms of detection and monitoring. So we have the early detection that we talked about, which is trying to catch an instance of a virus or a viral infection before somebody might themselves know that they're sick, and maybe let them know you are likely to be infected, so don't go out and visit your elderly parent or grandparent. We have at-home monitoring for people that are infected, so they don't need to go into a clinic unless there's a dire need. Also, we have the ability for public health monitoring. So there were some very interesting early studies of influenza-like illness detection out of Scripps that show that you can actually detect where and when flu outbreaks or influenza-like illness outbreaks were occurring based on the overall changes in smartwatch data in certain regions. And then finally, we can do hospital triage. So if we have information on a patient before they actually come in the door of the hospital, we have information on who needs the most critical care first. And finally, the goal would be to close the loop on this again with this just-in-time information delivery. So we could envision delivering public health information. So if there are large community outbreaks, rather than having to wait for information to come in about the results of the laboratory testing, we can actually get real-time information about people's physiology on a large scale. And then we can also deliver personalized health information. So for a person in particular to detect these changes in health or changes in physiology, that can then recommend them to come in for testing. So there are several studies going on in this area, and that the idea here I think is quite similar across these different studies, which is to be able to use smartwatches for either one or all of these areas that I mentioned earlier. So the studies I mentioned just previously were mainly using smartwatches. So these were the devices that I've talked about for most of this talk today. But there are also other types of devices that can be quite useful for detecting changes in physiology. And one example is this device out of Northwestern from John Rogers Group, which is a rubber patch that sits on the lower neck area, the suprasternal notch, and measures physiologic signals. So this can pick up information about the frequency, intensity, and sound of coughs and breathing, as well as respiratory rate and effort. And so you can imagine that this device can also be quite useful in monitoring for COVID-19. Although, again, the wearability of this is less than that of a typical watch, which is sort of more normal for people to wear than something that is a patch on their neck. So again, we always have this trade-off between the wearability of a device and the type of information that we can get from it. So I'll mention in a little bit more detail our study that's using smartwatch data to try to identify COVID-19 in individuals. And so this study has been funded by a variety of sources, and actually what's missing on this page is our support from the North Carolina Biotechnology Center, which has been incredibly helpful in keeping this study going. And we also have support from Microsoft Azure, which is supporting our cloud migration and our ability to expand this study. So what this study actually is a smartwatch study which takes information from both mobile devices and smartwatches and aims to learn who should be quarantined and or tested and when, and who will need specialized care resources and when. And our goal is to use information that we capture from daily surveys about people's illness symptoms, so whether or not somebody is experiencing symptoms, how severe those symptoms are, and whether or not they've been tested, and then using information from the smartwatch to try to develop digital biomarkers of susceptibility to infection, whether or not somebody is currently infected with COVID-19, whether there are pre-symptomatic or early signs of infection, or even asymptomatic signs. So although somebody may not feel sick, we may be able to detect that in fact they are carrying the virus. We want to understand the level of contagiousness and to be able to differentiate between COVID-19 and other illnesses that may look quite similar. And then finally, being able to predict the illness trajectory is very useful because it helps us to allocate resources. So participating in this study is quite easy. Everything is done virtually, so it's based on an e-consent. There's a baseline survey that gives us demographic information, and then we have information on COVID-19 and influenza testing. So that daily survey that I mentioned is 2 questions every day. This can arrive by email, text message, or in an iOS app. And initially, the study was designed to be every day for 30 days and then every week for 2 months. But as the pandemic continued, we actually found that we needed daily information because there was so much change happening so fast. So an overview of the platform, and I think that this was one of the biggest challenges in actually launching this study, was trying to build this platform as we were getting the study up and running. So the beauty of the way that we've set up this platform is that we're actually able to get 12 months of retrospective data from anybody who owned a smartwatch and wore it before they entered the study. So this gives us a very, very nice baselining period if we're to detect COVID-19 to be able to compare with past measurements. We've rolled out our study in many different languages, and we have lots of different technology choices. And the baseline enrollment survey gives us a lot of information about demographics, about people's typical habits, about whether there are comorbidities that would change the way we should interpret their digital biomarkers. And then over time, we are using information from the surveys to try to develop those digital biomarkers. And so this study then will go for 12 months from the time that a person enrolls, and we will be enrolling at least through March of this year. As the vaccine for COVID-19 comes out, we hope that numbers will decrease, but we're also interested in detecting other types of infections like influenza-like illnesses or influenza itself. And so this will continue to be useful past the life of COVID-19. And so you can see just a quick case study from one of the participants. We have over 6,500 participants in the study at this point. And of those who have had COVID-19, we can see that there are some clear trends. We see that sleep times tend to occur earlier. We have fewer awakening times during the night, but we have increased restless duration. And so this is information that was collected from a Garmin smartwatch. But our study can also take in data from Fitbit, Apple Watch, and any device that syncs with Apple Health Kit. So we see similarly with physical activity, these are two different people who have had COVID-19 infections, that we see a decrease in physical activity that is concomitant with the infection. And so what we've found in our study, in our early analysis, is coherent with a lot of the other similar studies in this realm. The Stanford study, which used an app that they developed called MyPhD, enrolled a similar number of participants, so roughly 5,262 participants. And you can see here the number of people that had been infected with COVID-19. The dark red are verified positive cases, and we have the dark gray that were sick but did not have a confirmed positive case, and then we have the golds that were sick but tested with something else. And so I think some of the interesting things to note about this study are the two metrics that were used as digital biomarkers to detect infection were the difference in resting heart rate. So again, that's that sort of change of heart method. And then also this other method that took into account a combination of heart rate and physical activity. So they call this heart rate over steps anomaly detection, or HROS-AD. And using each of these individually and in combination, this group was able to pretty accurately detect COVID-19 infections. And this method is based on some successes that we've seen on a larger scale, which the Scripps group had used to predict influenza-like illness. And this was pre-COVID-19, but the methodology is pretty interesting here, where there was the CDC influenza-like illness rate, and that's in red here. And they did this for 5 different states. And what you can see is they have 2 different models that they developed from Fitbit data from these different locations. So there were 47,000 people roughly who wore Fitbits consistently during the study period. And you can see that the models that were based off of the Fitbit data, there were 2 models. One had a lower threshold than the other, were able to really quite accurately predict what the influenza-like illness rates were in these different states. And so this same group, when COVID-19 hit, also developed a similar methodology. So you can see this is the user interface of the app that they developed. And again, they're working to, similar to these other studies, develop methods to detect COVID-19 from the wearable device data. There's another group that's out of Evidation Health that is also working on a similar area. So we can see that there's a lot of push toward this, and in general, the measurements that are used for all of these different studies are quite similar. So looking at the overall physical activity and seeing trends where physical activity decreases with the start of an infection, whether that is influenza or whether that is COVID-19 or some other type of illness. We see an increase in heart rate, and actually the increase in heart rate was less pronounced in the COVID-19 case than with the influenza case. And then also these sleep changes, so either increased sleep or increased disruption in sleep. And interestingly, this study breaks down the different types of symptoms and how these changes in physiology relate back to these different types of symptoms. So one of the things that when we think about the data in these studies, we find really important to consider is who the population is that are enrolled in these studies, because these were mainly all bring your own device models. And just to call out, in our own Cove-Identify study, where we really see a lot of the challenges, is in a bring your own device model study. So you can see that we're looking here at in blue, the national COVID-19 positive cases, and this is broken down by ethnicity. We see the national COVID-19 deaths, and then we see the participants in our study, and then the comparison to national demographics. And so what stands out, and this has been true from the start, so this data is from back in August, but since the start of the pandemic, we've seen that COVID-19 is disproportionately affecting people from Black and Hispanic communities, and that the proportion of COVID-19 cases and deaths is higher in these communities than their national demographics are. And even more challenging, that in the studies that we have where we ask people to enroll who already own their own devices, we see that the people who tend to enroll in the study and the people who tend to have these devices are in the white non-Hispanic category. So not those people who are disproportionately affected by COVID-19. And so this really can cause difficulties, as we talked about at the very beginning of this talk, that there are inherent biases in a lot of the technologies that are developed, and digital biomarkers will be no different. That when we use training data to train a model, that data is what teaches the model what to look for. And so in the case where we have far less data than is representative of the people who are affected by COVID-19, our models no doubt would be biased. And so one of the reasons actually that we have held back on developing our digital biomarkers and putting out information on our study is that we're working very, very hard to balance our racial demographics in our study so that we can be more confident that when we develop digital biomarkers, that those will truly work for all people. And so we've been working in a variety of different ways to address that. And we have a blog post out that you can read a little bit more about these efforts. But with Duke's CTSI CERI, or the Community Engaged Research Initiative group, we've had a lot of help with getting into communities that otherwise wouldn't have representation in our study. We've had a few grants and donors to help to support our ability to hand out devices, and we really hope that we'll be able to have a more representative distribution of demographics in our study so that we can confidently develop digital biomarkers that will work for everyone. So a couple of those efforts that I mentioned are here, and also we've been translating our study platform to many different languages. So we currently have Spanish, French, Polish, German launched, and we're in the process of launching Mandarin, Korean, and Hindi. And we're still continuing to apply for funding to be able to purchase wearables on a broader scale. So with that, I think that this quote from Topol and Oran is really key, which is that our goal with digital biomarkers, particularly in the case of COVID-19, is not necessarily to replace conventional diagnostic testing, but rather is to supplement conventional diagnostic testing, which is constrained by capacity, cost, and its one-off nature. And when we say one-off nature, that's because you might get tested today and then become infected tomorrow, and we would miss that. Or you might even be tested today and already be infected, and we might miss that. So we need these innovative tactics for public health surveillance. Crowdsourcing digital wearable data is one of those. There are methods that people have been exploring in monitoring sewer sludge, but there are all sorts of innovative methods that we can consider new technologies to enable if we think outside of the box and think about how non-traditional methods might help to give us more information about likelihood of outbreaks. So finally, with that, I want to touch upon the future of digital health and where we are hopefully going with everything that we've seen with this digital biomarker development and different sorts of wearables technologies. Engineers have been making huge progress with the ability to embed all sorts of sensors into novel materials. And so we've seen a range of what we might call wearable sensors, from something like a sticker or a tattoo that can be worn on the skin, all the way up to clothing that has embedded sensing in the fabric of the technology. And so what we see here is that wearables will someday soon be moving from maybe a wristwatch to something that is sort of fully sensing and is unobtrusive and not noticeable by most of the wearers. And so the benefit of this is that we can get continuous monitoring without having to sort of change our lifestyles or remember to plug in a smartwatch. So with improvements in battery technologies and with improvements in data compression and transmission and storage, we'll continue to see that we can get better and better sensing methods and more accurately, because we can get to locations on the body that we otherwise wouldn't be able to get to. So this is just one example of sort of a conductive tattoo type of material or a conductive ink that can sit on the skin. And then we have some examples here of the physiologic monitoring from the thread in a fabric. So it's just worn as a shirt, but it's actually sensing physiology. To facilitate our ability to collect data continuously, we need to be smart about how that data is pulled in and stored and how much data we actually collect, because what can be tricky is understanding the sampling frequency that's necessary for the digital biomarker development. So in this particular example, we were looking at what the minimum sampling rate is that is necessary to be able to reproduce parameters of heart rate variability that are clinically useful. And what we found here was that we could actually have the sampling frequency of one of our PPG devices and actually continue to have as high of an accuracy with heart rate variability as we would have with double that sampling rate. And so you can imagine that if we can decrease the sampling rate, we can decrease the amount of data that's needed to be stored, the amount of data that's needed to be transmitted. So this is an ongoing area of work for us and an area that we think will be very important as digital biomarkers become more and more prevalent. And we also want to make sure that there are easy ways for people in the community to be able to learn more about digital biomarkers and develop their own digital biomarkers if of interest. So we've developed this online platform and community called the Digital Biomarker Discovery Pipeline. And this platform is providing open source tools, algorithms, and resources to make discovering digital biomarkers more accessible and establish the best practices for the field. So I really encourage you to check out dbdp.org if you're interested in learning more about digital biomarkers. The platform itself has a variety of different modules, and so these are modules that have been developed not only by our lab but also by others in the community. We've collaborated with several other institutions that are developing their own pipelines for wearables and biomet data analysis, different methods for pre-processing, for reformatting data, for developing features from data, for performing feature selection and developing different prediction algorithms, or understanding the relationship between the wearable or biomet data and some outcomes of interest. And so you can check out these different modules. All of the code is open source on GitHub. And we have different code for different devices that are available. And we would really encourage anybody who is working in this space or interested in this space to please check it out and contribute your code, because these analyses and this code is useful when we all work together. And so rather than individual researchers and labs reinventing the wheel, we want to be able to learn from each other and to build upon each other's work. And there's a lot of documentation here, and so we're really focused on making sure that when people do submit their code or their data, that it is super well annotated so that somebody could pick it up 10 years later and still know exactly what was done. And so if you're new to the digital biomarker world and you want to learn more, we have a resource guide to help get you started. And I really encourage you to check this out and to become a member of the DBDP community. So finally, bringing all of this together, the vision for the future of healthcare is that we use digital data to empower patients to be able to understand their own physiologic information and to be able to put that into context based on their own past history and based on the general population. We also want to use this to develop precision therapies so that we know what is right for an individual person based on their own physiology, their own habits, and what their own needs are. And part of that is developing just-in-time adaptive interventions that get to the person right at the time that they're needed based on this contextual monitoring. And finally, what we can do here is improve access to care, because these devices, as we mentioned, are really quite low cost, are easy to design, and can bring a lot of information to people in an automated way. So the opportunity here for improving access to care is huge, and that's where we hope that most of our work is going. So with that, I want to thank many folks who we've worked with on a lot of the data presented here and a lot of the topics presented here. My lab members, and primarily I want to give a shout out to Peter Cho, who helped to organize the slides for this talk. We also have a fantastic group working on the COVIDentify study. And my student Brene Bent is the lead developer on the Digital Biomarker Discovery Pipeline, or DBDP. And we've gotten many grants to help support our work with the digital biomarker discovery, ranging from prediabetes digital biomarker discovery to infectious disease biomarker discovery. And with that, if there are any questions, please don't hesitate to reach out to me. My email address is jessilyn.dunn@duke.edu, and I'll be glad to answer any questions you might have.